Okay, thank you very much for the introduction, Tamás. We miss you a lot in the laboratory, and I hope I can convince you a little bit to come back to science, but let's see. Let's see later on. And I would like to thank to everybody for coming to this conference, and especially to Cytosurge, the sponsors, NanoSurf, for organizing this great event. So we learned a lot yesterday. And during my presentation, I will talk about the FluidFM, especially the Robotic FluidFM, and what we are doing in the Nanobiosensorics Laboratory. So I will shortly introduce our laboratory, so where we are located, what we did, what we are doing right now, why we needed the FluidFM, why we needed the robotic version of the FluidFM, and what type of research directions we were following with it. So especially I will talk about adhesion measurements, printing, and at the end of my talk, injection experiments. So basically our research institute is located in the Buda Hills. So this is Budapest city center, and we are here up in the Buda Hills, basically in a natural park. So we even have a nuclear reactor, a sports center, also we have the SAN data center, and so historically it was put outside of Budapest, a little far away on a safe place. And the Robotic FluidFM and our laboratory, the Nanobiosensorics Laboratory, is located in the building 26 at the second floor, and you are more than welcome to visit us. So please just drop us an email if you are in Budapest, and we are more than welcome to visit you and discuss common interests and ideas. And yeah, it's still working, but it's always a debate that whether we should stop it or we should keep it running, but it's still running. It's a research reactor. And so what type of research we are doing in the Nanobiosensorics Laboratory? So we are focusing on three main areas. One is the biomimetic surfaces, so especially surface coatings based on proteins, genetically engineered proteins, or hard materials like nanotubes. We are also very much in the field of single-cell biophysics. So our interest is developing techniques which are capable of manipulating and sensing, detecting single individual cells. And our main research area is the development of label-free biosensors, especially optical waveguide-based biosensors. And probably the concept is known for all of you. So when you have a commercial ELISA system, oh, sorry, maybe I have to be more careful with this. So sometimes it's disappearing. Okay, then yeah, a little closer. Thanks. Okay, so when you have a commercial ELISA system, so often you immobilize the primary antibody on a solid surface, and you have the antigens. You want to measure the concentration of the antigens, and you add a secondary antibody which is labeled. But in a label-free system, we want to get rid of the secondary antibody. We would like to monitor this binding event. So we need some kind of physical process which measures the binding itself. And all of the sensors we are using, this physical process is the refractive index measurements. So basically the adsorbed molecules increase the refractive index, and this is the signal what we are following. And the most traditional optical waveguide sensor is this OLS, which also you can find in the laboratory of János. So this is a very traditional old system. We have a high refractive index waveguide and couple light into the waveguide, and during this incoupling, this evanescent field is created, which can sense the biomolecules. And just like in quantum mechanics, if you want to entrap an electron into an area which has a size smaller than the de Broglie wavelength, you have discrete energy levels. And this is true for the photons. So if we want to entrap the photons in a layer which has a thickness of 1/200 nanometers, which is comparable to the wavelength, we have again discrete energy levels. So this means that only at one specific wavelength, the so-called resonant wavelength, we can couple the light into the waveguide. And by following this resonant wavelength, we can monitor the binding events. And this is the configuration which we developed with a Swiss startup company, the Creoptics. We combined the technology with optical interferometry and could reach superior sensitivity. So on the next slide, I show some data when we were monitoring the ion binding to genetically engineered proteins. And these are the kinetic curves of the ion bindings. And we could monitor the ion binding, and we could identify the low affinity, high affinity binding site, even at very, very low immobilization levels. So for example, when we immobilized 1 picograms of per square millimeter material. And this configuration we started to use in cell research. But not this interferometry setup, but we had a collaboration with Corning Incorporated, and we were using the so-called resonant waveguide grating principle. So again, we have the waveguide, the high refractive index layer, where we couple the light in. We again create this evanescent optical field to sense the biomolecules or the living cells. But in this case, the grating is so strong that when we couple the light in, it immediately reflected back into the direction of the substrate. So this configuration is especially suited for a plate-based screening, because all of the optics can be placed underneath the waveguide, and all of the liquid handling can be placed on top of the waveguide. And this was the configuration we initially started to use in cell adhesion research. So again, when the cells adhere on the surface, they simply deposit some material, so lipids, proteins, into this evanescent field, and this will tune the resonance, and by following the resonant wavelength, we can follow with extremely high resolution the adhesion event. And these are some real data I would like to show you. So we deposited a polymer which was grafted with the RGD motif. This is a minimal sequence for integrin recognition. And we tuned the RGD average distance in this experiment simply just by mixing the PLL-PEG RGD polymer with the PLL-PEG polymer. And these were the optical biosensor signals when the RGD distance was 5 nanometers, 16 nanometers, and roughly around 73 nanometers, the adhesion event disappeared. And this is a proof of principle study that with this configuration it is possible to monitor nanometer size events, and the cell adhesion disappears because the integrins need to cluster. And in this cluster, the cluster size is typically 100, 200 nanometers. It is not enough if only one integrin binds one RGD motifs. So it should be minimum 3, 4. And this is why if you have an RGD distance larger than the cluster size, then you don't have anymore the cell adhesion event. And we started to use this configuration in the researching of the cancer glycocalyx. We had some very interesting effects. We discovered quite recently that basically digesting some components of the cancer glycocalyx, we can reduce or we can initiate the cell adhesion. And this reduction is quite shocking. So even we can reach 60% of reduction of the adhesion event when we, for example, remove this chondroitin sulfate with some kind of enzymes. And also we continuously move to techniques which are capable of manipulating the cells. So one is where Tamás was working. So Tamás was working on the FluidFM, and also on this technique, the computer-controlled micropipette, which is a commercialized technique. A Hungarian startup company commercialized this technique. The idea is very simple. We have a glass capillary on an inverted microscope, and the robot moves this glass capillary. And when it moves the capillary above the cell, above the targeted cell, a negative pressure is applied, and we can easily pick up the cell. And just by tuning this negative pressure, we can measure the cell adhesion force, but in a very indirect way. So this was our problem, that this is an indirect measurement. So you know that with this negative pressure, we could lift up the cell, but we don't know the force. And also the resonant waveguide grating configuration we modified. So this is a not yet commercial system. So Corning never commercialized this system, but we could reach single cell resolution with the resonant waveguide gratings. So these are the optical biosensor signals when individual cells are adhering on the surface of the waveguide, and these are the spreading curves of individual cancer cells. So basically this was the starting point, and we were continuously looking at new technologies. So just to summarize, we were working on a large area. We wanted to manipulate individual cells, and we had this huge problem that either this technique or this technique, it cannot measure the adhesion force. So we don't know anything about the adhesion force. We know the optical signal here, and we know the critical pressure in the computer-controlled micropipette. And in that time, we were also focusing on the glycocalyx, the cell cycle, with using a cell cycle reported construct, and wanted also to measure these these population distributions of the cell adhesion strength. But we didn't have a force data. And then FluidFM came into the picture, especially with the robotized large area sample stage, the Omnium system. And we were very happy because this was a technique, it's highly needed for us and extremely ideal, because we could manipulate individual cells, we could directly measure the adhesion force of individual cells, we can even inject or sample individual cells, or use it for printing, which is also an important topic in single-cell biosensing, if you can print motifs for single cells or cell groups. So we decided to buy this technology and started the collaboration with Cytosurge back in 2019, 2020, when we started to use this technology. And the very first idea was to combine the technology with the optical biosensor technology. And for this, we created this sample holder for the optical waveguide sensors. So these individual squares has a size of 2 by 2 millimeters. So these are the sensing areas of the optical biosensor where we can monitor the resonant wavelength. So basically we have a picture something like this, and we wanted to make the experiment that we follow the adhesion of single cells and after move this microplate inside the Omnium system and insert the FluidFM probe, target exactly the same cell, and then we have the optical signal and the direct force for exactly the same type of cells. And we hoped that with this technology we can calibrate the optical biosensor signals to adhesion force. And the experiment was very successful. So you can see that these are the optical signals of a small, medium-sized, large area cell, and these are the force distance curves which we could record, and both the adhesion energy and adhesion force correlated quite well with the optical biosensor signal. And using this technique, we could calibrate the optical biosensor signal, and we could measure the adhesion force distribution in time for a large cell population, for hundreds of cells, which is very difficult to measure. And these are the population distributions of, which we recorded at 10 minutes, 15 minutes, and 85 minutes. So you can see that this population distribution of single cell adhesivity is spreading, and it has this characteristic log normal distribution. So this log normal distribution means that you have some kind of mean value, but even very, very far from this mean value, you still have a relatively large population of cells with large adhesivity. So this was a very important finding. And this is not even a primary cell sample, these were cell lines. And just to summarize the throughput, with a typical AFM, when you stick the cells on the cantilever of the AFM, typically one can measure the adhesion event of 5 to 6 cells per day. With the FluidFM today, we can go up to 100 cells, which is enough to get these population distributions. So I will show you data later on. But with the optical biosensor, with the calibrated optical biosensor, we can go up to thousands of cells. And the new addition, that this population distribution we can record in time. And why is it so important to record these distributions in time? Because please don't forget that this distribution is created by a power law function. So if you have one cell, the cell divides. From one cell, you will get 2 cells. And from the 2 cells, you will get 4 cells. From the 4 cells, 8 cells. So after n divisions, you have 2 to the power of n number of cells. And when I ask even the best students that how strong is this power law distributions, they really cannot feel it, how strong it is. Because if you get the 2 to the power of 300, so just only 300 divisions, that is such a huge number that there is not so much atoms in the whole universe. So this power law distribution is extremely powerful. So it can create a population distribution very, very fast. So I think the ideal technique would be to monitor these population distributions, even from a real sample, and monitor the change of these distributions in situ, in real time. But of course, this is far out of the reach of the present technologies. And we also use the Robotic FluidFM to calibrate the computer-controlled micropipette using this colloidal force spectroscopy arrangement. It was very convenient to work with the Omnium system, and we could conclude that the computer-controlled micropipette, which is measuring this critical vacuum, it can measure forces in the nanonewton range. So it is not so good like an AFM, which can follow adhesion force in the piconewton range, but the nanonewton range is enough to measure adhesion force of for example, completely attached cancer cells. So the typical force is in the several hundred or even thousands of nanonewtons for a typical adhered cell. So this calibration was also successful. So if you are interested, you can check these papers. And also we started to use the technique for biological problems. And the first problem we investigated was the cell cycle. So we used the Hella-Fucci construct, which has different colors when the cell cycle is progressing. And in the M phase, the mitotic phase, the cells are colorless. And this mitotic phase is a very, very important phase, because in the M phase, it was just recently discovered that the cells are adhered with these so-called reticular adhesions. So it was discovered in 2018 and was published in a Nature Biology paper. So these are not the conventional canonical focal adhesions. So this is very, very important to note. Even the protein composition is different in these adhesion contacts. And we were very interested about the adhesion force in these reticular adhesions. And by using the Omnium system, we could easily get the population distributions of the adhesion force, adhesion energy, and we even created a parameter which is the maximum adhesion force divided by the distance at the maximum adhesion force, which we call the spring coefficient of the cells. So basically it characterizes how stiff the cell is, and we also could measure the population distributions of this cell stiffness. And we discovered that from the biosensory signals we could see that these reticular adhesions are less dense than the normal canonical focal adhesions. But it was very shocking to see that when we divided the maximum adhesion force with the area, so the force per area was maximum in this reticular adhesion. So this is a very, very strong contact which is formed. And this was not proven before, but we could successfully prove this with the FluidFM technology. And also we were moving to more complicated systems using the Omnium setting. So we measure the adhesion force and cell-cell force of endothelial cells, epithelial cells. So we even put cancer cells on top, so creating this cancer invasion model. And the system was working fine. We could measure these force distributions. And typically, to get a reliable distribution, we found that we need to measure minimum of 30, 50 cells, which is out of the reach of the normal AFM measurements. So I think this is really the way to go when one needs to find the adhesion force distributions of relatively large cell populations. And also we use the technology for printing. So we printed cell adhesion motifs and fluorescent proteins on a hydrogel layer. And for printing, we were using these micropipettes. So this is the cantilever, and on the cantilever we don't have the tip, but instead of the tip, we have this 2 or 8 micrometer wide opening at the end of the cantilever, and using this opening, one can print very easily microstructures. And we demonstrated that we can print these structures on a huge area. So these lines are going in for centimeters. So we can print these micron scale lines or these spots for individual cells or for cell groups on centimeter scale areas. And the nice thing about the system that it can hold several probes. So it can be done that one of the probes you fill up, for example, with the RGD motif, and the next probe you fill up with another motif. For example, this pH RNS we are working, which is a synergy peptide for the RGD. And you can exchange the probe during the printing, and on the very same area you can print the RGD motif and print a controlled way the other peptide, which is very, very difficult to do with standard microfabrication techniques. And it's quite straightforward using the Omnium system. And what we are doing right now with the system, so I have 3 colleagues with me. Kinga has a poster on nanoparticle injection. So we are injecting various types of nanoparticles into the cells, for example, extracellular vesicles, and investigate of the fate of these nanoparticles. As you can see on the poster of Kinga, using the FluidFM technology, we can make such a configuration that this endosomatic lysosomal degradation pathway is not playing a role. Because, for example, if you have a normal uptake of the nanoparticle, that is immediately covered by a lipid shell. And very often it is going to the lysosome, and the cells try to degrade these uptaken nanoparticles. But when we are using the FluidFM technology, we don't have this. And this is proved. Please visit the poster. So we are thinking about targeting intracellular objects using this technology, which is impossible to do at the moment, because you always have this lipid vesicle around the uptaken nanoparticles. We are also working on injections of metal nanoparticles, and these are still ongoing works, but we could successfully got the nanoparticles aggregated inside the cell. So, for example, we injected nanoparticles with a size of 5 nanometers, and when they felt the local pH inside the cytosol, they started to aggregate, and they started to form some plasmonic objects, which could be used for sensing. I think it can be interesting in specific areas, because with the sampling you can get out the materials, so you can see the biomolecules inside the cell at very well-defined time points. But if we can create a sensor inside the cell, so a plasmonic sensor inside the cell, and it's covered with some kind of antibodies, then we hope that we could continuously monitor the concentration of one type of protein. So this is our aim. We are working on it. And the two other colleagues of mine, they are working mainly on adhesion research and focusing on the glycocalyx, so degrading the glycocalyx with some enzymes and measuring the adhesion and the population distributions of the cells when the glycocalyx is degraded. And we are seeing very, very interesting things. So for example, when you start to remove one component of the glycocalyx, it's not only the population distribution which is moving. We create all the time subpopulations. So in many cases it happens that we create a subpopulation with very, very low adhesivity and another subpopulation with very high adhesivity. So if somebody is measuring the averaged adhesivity with another technique, which, for example, averages the signals of thousands of cells and measures that this is the averaged adhesivity, the averaged adhesion force, maybe there is not a single cell close to that adhesion force value in the population, because you have these two large subpopulations. So I think this is very interesting, and we would like to explore these ideas with the Omnium system. And we are also thinking about printing some motifs for single cells on the biosensor surfaces, and very, very, very interested to go into the extraction experiments. So 2 years ago, we created a PCR laboratory. So we have the equipment, we have the knowledge, but unfortunately, we don't have the knowledge to make the single cell screening. So we are not good at in PCR in that level, but we hope that in the future we could make this sampling, maybe study from you how to do this, and we could reach the single cell resolution. What right now we can reach is that if we find a subpopulation in adhesivity, we can select, for example, 50 cells from that subpopulation, and we can run a PCR on that subpopulation. So this is the level where we are right now. And I would like to thank very much for Cytosurge and the companies for their helps, all the members of the Nanobiosensorics group, especially those who were working on the FluidFM system. I thank also to NanoSurf. So we also have a NanoSurf AFM, and we are thinking about to buying the standalone FluidFM add-on on the NanoSurf AFM and do some electrochemistry with the cells, because we have a very, very strong group in electrochemistry in the institute, and also a very strong group on graphene research, and this might be useful. And I would like to thank to János, Professor János Vörös, who basically initiated these ideas, and he suggested me to go into this field. And thank you very much for your kind attention.